Good afternoon, all. I am Claudio CONCI, a biomedical engineer from Politecnico di Milano, and today I'm going to talk about something a little different. So it's quite a new method or a new device to image the in vivo fibrotic reaction. So we start with the biocompatibility assessment of biomaterial or new drugs in general, which entail an inflammatory event primarily reflected in cell behavior at the microcirculatory system, basically. The current protocols used in preclinical studies are based on histopathology, but these protocols are analyzed over 40 years old and require the sacrifice of a huge number of laboratory animals, with, as you may know, an unsustainable ethical burden of animal research. So in this context, as to optimize the validation procedure and at the same time to reduce the real cost on approval of, for example, new biomaterial drugs or vaccine, several attempts were done to introduce intravital microscopy techniques as a standard protocols to quantify the subcutaneous inflammatory reaction. And this is done in terms of neovascularization rate, fibrosis as the generation of collagen type 1, and cell infiltration. Therefore, by using intravital microscopy, we can potentially study the implantation outcome in a real-time manner. But no device right now can provide a specific tracking geometry to reposition the field of view of your own microscope for repeat analysis in the same animal and in the same spatial region, not basically allowing to consistently replace the histological ex vivo analysis, which are the standard right now. In addition, the quasi-totality of the employed imaging windows present invasive metal frame, as you can see on my slide, and a persistent subcutaneous access for the whole experiment. Then, to reduce the invasiveness of that techniques, researchers recently introduced the use of scaffold as a micrometric imaging window implanted under the animal cute, just to study the progression of the foreign body response, ectopically guided and confining the tissue response in the desired region of interest, and then providing an optically accessible field of view. Then, in this context, I reintroduced an innovative miniaturized scaffold-based imaging window, which was designed for intravital nonlinear microscopy. We call that device the Microatlas, and is implantable subcute in living mice, or, as you can see on the left-hand side, in contact with the chorial on-top membrane of living chicken embryos. This device is inspectable without the need of a persistent percutaneous access, and allow translational studies over time of the same animal and the same region of interest. The Microatlas, differently to other windows, is fabricated by two-photon polymerization, and we used the biocompatible organic-inorganic SZ2080 photoresist. It's quite common right now. Again, there is feature size goes beyond the Abbe diffraction limit, so we reach a feature size of the Microatlas Stratis down to 100 nanometer. And is enabled to recreate a 3D microenvironment for cells and offer a truly 3D adhesion site similar to those present in physiological tissue. So I devised a device able to potentially guide an angiogenesis into an animal to allow repeated intravital imaging of the same animal too, and offers atlases for microscope repositioning and a structure suitable for adaptive optics, just to correct optical aberration. Just take a look to the device. The device was realized on circular glass cover slip from the dimension of 12 millimeter down to 5 millimeter, as you can see on the left-hand side. The device is constituted by several scaffolds on the, as you can see in the red square, and each scaffold is constituted by a pore size of 50 micron per 50 micron with a height of 20 micron. We superimpose 5 layers, obtaining a scaffold having a square side of 500 micron and a total height of 100 micron. We can see in the far edge of the curved slip some spacer as to allow the position of the microatlas in the correct site onto the animal, and we can find some planar landmark as the axis coordinate axis, which allow you to reposition the microscope, or vertical landmark you can't see because it's too small, but in the central part of the SAM image you can see a small cone that allow you to focalize on the correct vertical plane. So I validate my device feasibility in living chicken embryos by performing the well-known correlontoic membrane assay. So I implant the Microatlas at incubation D7 of the embryo, where the cam is quite formed, and I conclude the observation rate at the 30 days of the embryo development. I first quantified, by using confocal fluorescence microscope, the cell density inside each scaffold, comparing my data with untreated samples. So the left-hand side of your slide is the control. As you can see from the graph below, results show a stability in cell density in control region, given no significant difference in all the experiment time point. In contrast, the infiltration in the Microatlas reached a comparable density with respect to untreated embryos. So at the third day of implantation, and exhibited a strong cell repopulation just after 48 hours of implant. In addition, cell infiltration inside the microscaffold showed a continuous growth till the 7th day after implantation, where the final reached density was more than 1.5 times more or less greater than untreated samples. In addition, as we can see from the confocal acquisition on the right-hand side, we can clearly state that tissue remodeling aimed to angiogenesis is giving rise to microcapillars inside the Microatlas scaffold. Then we imaged label-free by exploiting two-photon emission fluorescence microscopy our samples, just to try to quantify the foreign reaction as we can perform with histopathological studies. So we analyzed cells and neovascularization with the intrinsic autofluorescence of the tissue and the fibrosis, observing the second harmonic signal of the collagen generation. So then, in this regard, I propose data regarding 7 days of implants on the last time point, and what I observed into the Microatlas, as you can see on the left-hand side, where I segmented vessel both on control and Microatlas, observed a pronounced capillary microvascularization density 6 times greater than untreated samples. Then, if you move on the right side, second harmonic generation signal also collected in vivo highlighted the deposition of collagen type 1 inside and nearby our scaffold. By quantifying the collagen 1 directionality, I obtain an almost constant fiber orientation inside all the colonized Microatlas scaffold volume, irrespective to the computed axial coordinate, with a gradual decrease as that fiber exits each scaffold. This probably indicates that my scaffold architecture can influence the matrix formation. In contrast, untreated embryos show the sparse fibers directionality distribution with a significant difference with respect to the implanted side. Finally, by analyzing the collagen 1 matrix, the position density in planted sample did not exhibit a significant difference in the secreted matrix amount with respect to untreated embryos, thus showing a similar capsule density between the two cases. So basically, our device probably is not being encapsulated inside our living embryos. So in conclusion, I precisely characterize fabrication process and parameter to realize this miniaturized imaging window with a 3D scaffold that can be implanted in vivo into living organisms. The presented promising results highlighted the efficacy and reliability of the Microatlas platform. Its unique feature to encompass in a square millimeter area a single microfabricated structure that has a number of advantages from the point of view of tissue engineering and of intravital imaging. From the tissue engineering point of view, the Microatlas is actively colonized by the host tissue and allows for angiogenesis and does not seem to elicit a massive fibrotic reaction. from the point of view of in-vivo tests through optical imaging, the Microatlas do its autofluorescence under laser excitation, allows for sample repositioning in the microscope field of view and for long-lasting longitudinal studies, reducing both invasiveness implantation and imaging protocols. So my future purpose is to exploit these unique features to devise implantable micro devices for the test of inflammatory reaction to biomaterial in rodents, or for the test of drugs or vaccine on chicken embryos, giving a strong impact, hopefully, on imaging window development and giving rise to new device which can incorporate atlases for long-lasting observation and scaffold for tissue engineering and regenerative medicine. So thank you all. I want to thank you in particular my lab mate from Politecnico di Milano, led by Professor Raimondi, and from Università di Milano Bicocca, led by Professor Gisberto Chirico. And thank you for your kind attention. I hope to see you next year in person with the FOM. So thank you.